Hello everyone, welcome to Dr. K. PREM-PRIMER video lecture series presenting by Dr. K. Prem. That's me. In today's lecture, we'll talk about DNA cloning. So you can see the definition of DNA cloning here. Production of same copies of DNA fragments or genes using genetic engineering. So when you want to clone any gene, right? Why you are cloning that gene? Because you want to get more copies of that so that you can go for sequencing, or you can go for in vitro mutation, or you can express that gene. Basically, you need a lot of copies of the same gene. So for that you will clone. Yes, you can see here the definition: production of same copies of DNA fragments or genes using genetic engineering. You are producing the same copy, exact same copies, right, of DNA fragments or genes using genetic engineering. That technique is called as DNA cloning. Right? Production of same or exact copies of DNA fragments or genes using genetic engineering is called as DNA cloning. In today's lecture, we'll know what are the steps involved in the DNA cloning and the various strategies of DNA cloning. DNA cloning, any DNA cloning has 4 essential steps. Any DNA cloning has 4 essential steps. They are: the first one is isolation of DNA fragments or DNA fragments isolation, or nothing but genes isolation. The second step is joining of DNA fragments and vectors. Once you isolated the DNA fragments from the genomes or any other source, then you ligate those DNA fragments with the vector vehicle. That's a vehicle, so that will your gene of interest is ligated into the vector by using ligation process. And third process is, once the vector and insert or DNA fragments are ligated, such molecules are introduced into the host cell. That step is called as introduction into the host cell. Why do you do? Because those molecules are, DNA molecule with the vector and insert, or vector and the DNA fragments are introduced into the host cell to make many copies, of the same copy. Third one is screening of recombinant clones. Once you transform your vector insert containing DNA molecule into the E. coli or phase, right? So they'll form that vector is having genetic selection marker. So that genetic selection marker may be an antibiotic selection marker or auxotropic marker. That auxotropic or genetic selection marker give a specific trait to the host cell. Based on the trait given by the vector to the host, you will screen. First, you select the transformants, then followed by you go for recombinant clones. So the fourth step is screening of recombinant clones. Here, I'm again repeating the same thing. DNA cloning procedure has 4 important steps. The first one is isolation of DNA fragments. The second one is joining of DNA fragments and vectors. Third one is introduction into the host cell. And the fourth one is screening of recombinant clones. In the next slide, we will see what are different methods for each step we have, right? So you can see again the DNA cloning steps, isolation of DNA fragments. The second one is joining to vector, and third one is introduction to the host cell, and the fourth one is screening and selection of recombinant clones. So here, what are different methods or routes available for each and every step? There are 4 steps. There are 4 steps which are there in the DNA cloning. The first one is isolation of DNA fragments. The second one is the joining of DNA fragments with vector. Third one is introduction into the host cell. And the fourth one is selection and screening and selection of the recombinant clones. For each and every step, there are more than one way of doing it, right? So when you look into the isolation of DNA fragment, that's the first step. Let us look into the what are the available ways to get the DNA fragments. The first one is restriction endonuclease. You are isolated at the genome. That genome is digested with the particular restriction endonuclease. Then you get a DNA fragments with the cohesive ends. So you isolate the DNA from any cell or organism, then you subject to the restriction digestion with your desired and restriction nuclease, then it will produce the DNA fragments with the cohesive ends. So that's how you can isolate the DNA fragments or genes. The second one is mechanical sharing. Mechanical sharing. Here, this is a physical method of DNA fragments generation, where isolated pure DNA is subjected to the sonication. Subjected to the sonication, you get a certain length of DNA fragment. So it may be a 2 kb or 4 kb or 10 kb, right? Those are blend ends. The first one is an enzymatic digestion to get the DNA fragments. Second one is a physical method to generate the DNA fragments. Third one is duplex cDNA synthesis. The mRNA which is isolated from the cells is converted to the cDNA by reverse transcription. Reverse transcription. So your mRNA is converted to the double-strand cDNA molecule by reverse transcription by using the reverse transcriptase. So you get the blend end DNA fragments. That's a cDNA molecules. And the fourth one is direct chemical synthesis. If you know the sequence of any gene or any DNA fragment, that entire length of that gene or DNA fragment can be synthesized in laboratory. This is a purely chemical method. So first one is a enzymatic physical method. This is a biological method. And this one is a chemical method. Chemical synthesis of entire gene or a DNA fragment when you know the sequence of it. And the fifth one is in vitro amplification of a DNA. So you have a template, right? You design a primer, 2 primers. That primers are specific to your gene of interest. So these 2 primers, after denaturation, they will anneal to this specific region of your gene. Then by using the Taq polymerase, you will produce the millions of copies of the same gene. That's called as in vitro amplification of desired gene by using the 2 primers. That's nothing but polymerase chain reaction also produces the DNA fragments or genes. So these are the 5 different methods we have to isolate the gene fragments or DNA fragments. The first one is restriction endonucleotidization, mechanical sharing, duplex cDNA synthesis, direct chemical synthesis, and PCR. So next step is in DNA cloning is joining to vector. How do you join your isolated DNA with the vector? So what are the available ways to do it? Let us see. The first one is homopolymer tailing. Homopolymer tailing useful whenever DNA fragments have a blunt end. Blunt end. When they have a blunt end, they cannot be readily ligate with any known vector. So you can add homopolymers, that is a poly A or poly T, poly C or poly G to the insert by using the terminal deoxynucleotide transferase. Then complementary polynucleotides are added to the vector. Then you can ligate them. You can insert or DNA fragment and vector can be ligated. You can anneal them and you can transfer into the host. Then you get a recombinant DNA molecule. That's called as homopolymer tailing. The next one is cohesive end termini. Your DNA fragment have a you digest the one DNA fragment with one enzyme. So for example, EcoR1. The same enzyme you use to digest the vector to linearize, then the vector and insert have a compatible cohesive ends. Compatible complementary cohesive ends, then the ligation process is very easy and efficient also. So the second method is cohesive end termini ligation. Third method is blunt end ligation. Any DNA fragment which is having the pi' phosphate and 3' OH, and other DNA fragment with the pi' and 3' phosphate and 3' OH. So the T4 DNA ligase can mediate the both the cohesive end ligation as well as the blunt end ligation. So if you ligate the vector and the DNA fragment and T4 DNA ligase, and then you see there is a phosphodiester bond formed between the DNA fragment and the vector. That's called as a blunt end ligation. This is a bit tough to have a clones through the blunt end ligation. You don't get many clones through the blunt end ligation. And the linkers and adapters. Again, I'm telling you, whenever you have a DNA fragments, maybe a genomic or cDNA molecules, which are blunt-ended. So you can add linkers. The linkers are the short oligonucleotides with the internal restriction site. So you can add linkers to the blunt-end DNA fragments, then followed by the restriction digestion that leads to the formation of cohesive ends. Those cohesive ends are compatible to the vector and they can be ligated. And adapters are also oligonucleotides which are free-formed cohesive ends. They have free-formed cohesive ends. So adapters, if you add to the blunt-end DNA molecules, and they have a free-formed cohesive ends, and they are readily ligatable with any vector. And the TA cloning, the last one is a TA cloning. When you amplify any DNA fragment by using the PCR, Taq polymerase have an ability to add adenine at 3'. Every DNA fragment you get through the PCR have adenine at 3' end. Adenine at 3' end. If your actor is having the free T, and your insert, through the PCR, DNA fragment you get from the through the PCR have a So A and T are complementary. They can be ligatable. So that strategy is called as, that method of ligation is called as joining of method is called as TA cloning. Once you ligate your DNA fragment with the vector, and you should introduce that into the host cell, so that you can get the enormous copies of the same construct, right? For that, you have to go for third step, that is a introduction into host cell. There are three methods in this step. One is the transfection with the recombinant phase DNA. Transfection with the recombinant phase DNA. Transformation into E. coli with the recombinant plasmid. And third one is in vitro packing. Transfection means that when you take lambda phase vectors, for example, lambda phase vectors. So maybe replacement vectors or insertion vectors, you clone your gene of interest into the lambda phase vectors. So those lambda phase, once you get that ligation mixture, if you incubate with the E. coli, then that is absorbed. So E. coli, lambda phase DNA, recombinant lambda phase DNA, if you incubate with its host, then it is injected into the host cell. That is what is called as a transfection, and it forms the plaques. Then transformation. So the ligation mixture which had insert and the vector, which is both are ligated, transformed with the competent E. coli cells. Competent E. coli cells, so which are treated with the calcium chloride, they can take up the DNA from the external source, and you can select the, then that's what is called as a transformation. In vitro packing. In vitro packing means, for example, cosmids. If you use the cosmids as a vector, you clone your gene of interest in the cosmids. Then cosmids have a cos sites, and you provide the tail and head and the other materials to that recombinant cosmids, then they'll form a phage. That is called as a in vitro packing. In vitro packing. So these are the three different methods to introduce your DNA molecule with the insert. And the fourth one is screening and selection. There are different methods are available for this one. The first one is hybridization, and the second one is PCR. Third one is immunochemical, and the fourth one is protein-protein interaction, functional complementation, gain of function. These are the almost 6 methods are available to screen the recombinants. The recombinants or transformants which provide some trait to the host cell. So that host cells are selected, and after that if you have a probe of your gene you cloned, you can go for the hybridization. You take those colonies are subjected to the replica plating, then followed by the baking with the nylon membrane, and flood with the probe. Then you see radiolabeled probe. Then you can see the recombinant clone from out of the thousands of clones. Then PCR. So you have 100 colonies. Pick up the smallest possible biological material from each colony, and place them into the PCR tube. Add primers to that, and go for the PCR. Whichever tube have a amplicon, those are positive colonies. You can go back to the colony, and you can select them as a positive colonies. Immunochemical. If your gene of interest has protein, that the protein has already used for the raising antibodies, those antibodies can be used to screen the gene of interest which is expressed. And the protein interaction, certain proteins have strong interaction with the other protein. Then we can pull out those colonies by using those protein molecules. Functional complementation. If a gene has established function, so if you have a mutant of particular gene, and that related genes can be cloned and transformed into those hosts, and the host behave normally, means your gene has that cloned gene is functionally active. So gain of function, any enzyme encoded or transporter encoded gene is cloned, then you can check for the activity of the enzyme or transporter. Then you can say that those colonies are positive or recombinant clones. So this is the overall view of DNA cloning steps and other methods or routes available for each and every step of the DNA cloning. You can see here, if you digest your DNA fragment genome with the restriction endonuclease, then that will have a cohesive end termini. And the cohesive termini can be ligated with the cohesive vector, and you can go for the transformation into the E. coli. And if you have duplex DNA, that's a blunt end, you can do the homopolymer tailing, then you can go for the joining of the vector. And then you can go for transfection with the lambda phase vectors. And if you have direct chemical synthesis, you can add linkers or adapters, and you can go for in vitro packing. If your gene is synthesized by the PCR, then you can go for joining of the vector with the TF cloning means, and then you can go for transformation. And you can choose these methods to screen and select the recombinant clones. Right? So this is all about DNA cloning. Hope you like my lecture. If at all, you please try to subscribe my channel. That's a Dr. K PREM-PRIMER for many updates. If you wish to share the content, no issues. You can share. When you like it, try to give thumbs up. And if you have any comments or clarification, if you need any clarification, just write to me through the comment section. I'll get back to you once I see your comment. Thank you all for listening. See you again. Until then, bye-bye.